Okay, so I did my presentation on proteomics. Proteomics is a field that's emerging in medical cell biology, especially, and it can be used for things like the early detection of cancer and detecting other sort of ailments that you can find that might pop up in the human body. So this is definitely going to be a very um, important field that needs to be studied in the future if we're going to be able to improve our medical quality. So just to start off, what exactly is proteomics? If you put it simply, it's the study of proteomes, but what does that mean? So a proteome is the complete set of proteins found in an organism, an organ, or a system. So you could take something like the proteome of the entire species Drosophila if you wanted to. It would be a really long process, but you could do it. Or you could take a proteome of an individual, or you could even scale it down even more. You could do just the proteome of a liver, a human liver, if you wanted to. The thing about proteomes that separates them from things like genomes is that proteomes are not static, so they can vary between cells, between organs, between individuals, and obviously between species. And they also tend to vary over time. So you're going to be producing and excreting different proteins at different times, and in that way, this is indicative of the underlying transcriptome. So as you can see here, we have this kind of scale of this kind of scale of the nomes, if you will, the omics. So you have the genome, which codes for the transcriptome, which codes for the proteome, which produces parts of the metabolome. And this chain is extremely important to study with all of them. Obviously, the genome is extremely important, so we can see genetic factors, but the proteome is important too because there are things like epigenetic factors that affect things where it's not necessarily going to be noticed in the genome. So why study proteomics? So it gives you an accurate picture of protein activity. So you can actually look at it if you have a baseline for what the proteome is. Then you can say this is working fine or this is malfunctioning. Genomes are relatively constant, like I said, but proteomes are dynamic. So you can see how the body is changing over time and compare whether or not that's a healthy thing. Another thing is that transcriptomics with RNA analysis, they're not necessarily correlated with proteins because mRNA doesn't always translate to proteins. So proteomics is a more accurate way to look at things that transcriptomics can look at as well in real time. Another thing is that proteomics can detect the presence or absence of a protein and measure the quantity of the protein, and that's really important in things like where there is you have too much of a certain protein, or you don't produce a certain protein at all. So there are lots of methods for studying proteins. Um, the first one is the Edmond degradation. So you essentially degrade the peptide in steps to determine the complete chemical composition of it. Newer ones are going to be mass spectrometry. Mass spectrometry, you use a centrifuge to separate each of the molecules out by mass, and that's used to characterize the protein and identify it. And then finally, there's immunoassays, which use antibodies and antigens to detect the proteins. And so those are going to be the top two. Those are going to be the main ones that people who are studying proteomics are going to be using nowadays. So proteomics, they allow us to study a lot of different things. So for example, how proteins are produced or what proteins are produced and how they degrade, how quickly they degrade. Um, there are post-translational modifications that are extremely important that we need to study. So after RNA translates a protein, they are occasionally modified, such as phosphorylation, which is very important in the activation of enzymes. Um, there's also things like protein-to-protein interactions that are extremely important. So prion diseases like Creutzfeldt-Jakob disease, those are going to be essentially a prion is a misfolded protein, and it causes other proteins to misfold through these protein-protein interactions, and it pretty much destroys your entire brain. So it's extremely important that we can actually learn how they cause this, and we can actually seek to prevent it. And the same with Alzheimer's. Alzheimer's is a big protein to protein interaction that obviously affects millions of people, you're in the world. at any given time. So it's extremely important that we study this in order to try and reduce the deaths related to Alzheimer's. We can also look at how proteins are expressed, when and where, and that will help give us an idea as to whether or not something's abnormal or something is normal, which is extremely important in obviously studying whether people are doing well, doing okay if they're healthy. And finally, we have biomarkers, which we can use possibly to detect diseases, such as cancer. So, for example, there is a prostate-specific antigen that is expressed when someone has prostate cancer, and it's extremely if we can just use that, if give someone a urine sample, and they can test for this prostate-specific antigen either in the urine or blood, that's going to be a lot more accurate of a test for prostate cancer than the normal prostate exams. And I know most men would prefer not to get a regular prostate exam. So in the future, proteomics hopes to do things like in-depth analysis of systems at the cellular level. So hopefully they can analyze how disturbances at the cellular level affect protein production in the entire system. So that includes things like cell cycle, how cells differentiate, and how carcinogenesis affects the actual cells. And that's going to be obviously extremely important in medicine, stem cell biology, and evolutionary biology. And finally, the human plasma proteome is something that's currently being worked on. So essentially, human plasma, which is the liquid that contains blood cells, it contains tissue linkage proteins from all parts of the human body, and that will give you current information on all tissues essentially. The problem is, at the moment, it's difficult to interpret because protein turnover widely varies between proteins, which makes understanding how they interact over time and how things work over time, what a good baseline is, makes it difficult. And then further, abundance is wide ranging between proteins. So you'll have a lot of one protein, a little of another, a medium amount of a third, and that makes quantitative understanding difficult. So that sort of thing is something that we're going to have to work on to develop in the future. But overall, I think that all of this is going to greatly improve our quality of life and how well we detect diseases in the future. And we've got this NASA video. I would recommend all of you watch it because it explains it a lot better than I do. So make sure you watch that. And yeah, does anybody have any questions?